Here is the home page for CBioPortal. If we want to look up queries for certain genes, here you can do it directly from the home page. So depending on the gene alterations you want to look at, you can type in certain genes here in the box. One of the genes that we know is most commonly mutated in cancer is P53. But you need to remember that you need to enter the exact gene symbol. So for P53, it is TP53. And you will notice here that you'll get a check mark if you have entered in a correct gene name. Now the CBioPortal has a list of all the different cancer studies that have been published. Now we can look at TP53 mutations across all the studies or looking at certain studies. And we'll start off looking at P53 alterations from all the cancer studies. And we'll be looking at both mutations and copy number alterations. So once you're ready, you can click Submit. And you will see a chart listing the percentage frequency of P53 mutations over all of the different studies. Now, if you want to know the identification of each study, you can roll your mouse over one of the lines, and here P53 is most commonly mutated in this ovarian cancer study from the TCGA network published in 2011, which had 94% mutations of P53. And looking at the second most commonly mutated form of P53 was found in this uterine study from the TCGA. So if you want to look at specifically a specific study, you can click on one of the lines, and here we'll click on the ovarian cancer line. And here we can see the P53 mutations and how they're mutated from all of the patients in this study. And you see, and since P53 is mainly downregulated cancer, it's not surprising that we only see 2 of the patients that have an amplification for P53. Here in black, it's a truncated mutation, and in green, here we see a missense mutation. So let's say you're interested in looking at specific patients. Here we can click on one of the lines, and we can see an overview of this specific patient. And not only see mutations not only for P53, but also other mutations that was seen in this ovarian cancer patient. And what's also available is actual tissue images from the Cancer Digital Slide Archive from each of the patients. So right now we'll go back to this chart that lists all of the studies and the percentage of P53 mutations. And let's say you're only interested in looking at a couple of the studies and looking at P53 mutations. You can click on Customize Histogram and then select Studies. And right now all the studies are listed. Here you can click None. And then let's say we want to look at the 2 acute myeloid leukemia studies. Then you can scroll to the bottom, and here it's showing the mutation frequency of P53 for the 2 AML studies, which we see is around 8%. And let's say if you want to print this data, either for the 2 studies, or let's say if you want to go back and look at all the studies together, all you do is click on the PDF link. And here, depending on the type of computer you have, you can download this file showing you the alteration frequency of P53 over all of the studies. Now, let's say if you want to only look at data sets looking at specific cancers, you can click on the dataset link at the top of the page, and here it lists all of the different data sets from the TCGA project that have been published. And let's say if you want to again look at the acute myeloid leukemia, You click on the study, and here it'll give us a study summary of different things like overall survival, disease-free survival, as well as different areas comparing gender, disease-free status, and if they've had a transplant, as well as also looking at the mutations in the genes and percentage of the genes that were mutated, as well as if any of the copy numbers of the genes were altered. Now, let's say if you only want to look at comparing females who had AML compared to the overall population, you can click on the blue part of the pie chart for the female, and that is listed here in red here at the top, so it can compare survival as well as the mutation rates comparing females versus the overall survival. It can also see the split between the number of patients if they've had different transplants as well. And you can also here see the list of the mutated genes here in the study and how many mutations that were available. So this is a great service if you want to see overall data with these genome studies that have been compacted into these relatively easy to read charts. So say if you want to look at a different study or in a different cancer, if you want to look at one of the breast invasive carcinoma studies, and here we'll again receive a study summary, including again overall survival, disease-free survival, as well as what we saw with the AML study. You see gender. Here in this study, 996 are female and 10 were male, as well as different categories such as race, ethnicity, where the tissue came from, as well as the stage that the patients were staged at diagnosis. And if you scroll more towards the bottom, here's a nice bar chart seeing how many of the patients and what age they were diagnosed. So you see most of the diagnosis here with this breast cancer study was around between the age of 50 and 70. And you can also see the different mutation counts as well as the overall survival in months and disease-free survival. Here we are at the home page for the Cancer Genome Atlas Research Network group. Now, if you want to download the raw data from the different genomic studies, here you can click on Launch Data Portal at the top right-hand corner of the page. And it will take you to the portal overview of all the different cancers that have patient data from the TCGA network group. So all you need to do is click on the cancer that you are interested in. So right now we'll click on the acute myeloid leukemia group. And you see right now they have a total number of cases set at 200. So for the data you want to look at, you can separate by the different mutation data that you're interested in. Right now I'll click on the total cases. And here if you scroll down, so here's listed all of the different types of genome mutations from the different institutions that are part of this AML group, as well as the name of all of the different patients in this group. And you can scroll down and see the name that's listed for all the different patients. Now TN stands for tumor, a sample that has a tumor as well as a matched normal control. And here the levels are listed as 1, 2, and 3. Level 1 is if you're interested in raw data that has not been normalized. Level 2 is normalized single sample data. And level 3 is interpreted data where, for example, you can have expression signal of a gene for a specific sample. So for this query, I will be looking at level 3. And depending on the different mutations you want to look at, let's say if you're interested in methylation, and if you're interested in the patient samples from Johns Hopkins University, you can click on either individual patients, or you can click, if you want the whole level, you can click the whole level. But let's say for right now we're only interested in certain patients. You can click on what you're interested in, and once you're done with all the data that you need, click on Build Archive. That's at the top left-hand corner of the page. Here, you'll need to enter in your email address, where you'll receive an email for the link of the data that you've requested. And once you download the files, they're in a tar file that will be expanded to include text of the data that can be read on your computer. And one thing that you need to note is that the links are only good for 24 hours. If you need to go back and look at your query and it hasn't been downloaded, all you need to do is go back to the website and repeat the query that you need, and you'll receive a fresh link to the file for the data that you're looking for. And what's also important to remember is you need to remember the publication and presenting guidelines for the TCGA network. And there are some restrictions if the paper by the TCGA project for the certain analysis you're looking at has not been published yet. You need to check that before you present or publish your data. Here you can click on the publication guidelines, and here it gives you an overview of the guidelines, as well as if the data stats where you're available to present the data. So here for AML, there are no restrictions. All the data is available without limitations. Here, if you scroll down to the bottom, you see where some of the cancers, like esophageal cancer, the publication's limitations are in place until July 7th of 2015. And if you want to make a different query for different data sets, all you do, you can go back at the top right-hand corner again, click on the Launch Data Portal, and you're taken back to the page of all the different cancers. So if you want to make a different query for data, you can click on the data that you are looking for.